of the conversion, at first I want to ask a question. Why pyruvate convert to acetyl-CoA?
Because acetyl-CoA will unite with the oxaloacetate and
No, not oxaloacetate. Please imagine the Krebs cycle.
So what's the situation? Krebs cycle.
Krebs cycle.
acetyl-CoA is a ketoglycine.
Also can enter actually to the Krebs cycle. I want to talk about the regulation. As I mentioned, whenever, what is the actual output of the Krebs cycle? Energy. Energy. In a simple word. Krebs cycle produce the energy. So whenever you have ATP, it means that energy level is good. So it's considered as an inhibitor. You know, whenever you have a lack of energy, you are so tired, okay? Lack of energy, pyruvate converts to acetyl-CoA to enter the Krebs cycle. So ATP, it means that everything is good. High energy molecule. NADH, high energy molecule. And also acetyl-CoA, because this is a product, it can inhibit the enzymatic reaction. But about activator, ADP, it means that you are in a hungry state. So pyruvate convert to acetyl-CoA, so it's entered to the TCA cycle or Krebs cycle. And also NAD plus consider as a low energy. And substrate, this is a substrate, CoA, coenzyme A, consider as an activator of the pyruvate dehydrogenase. What about the second session of the carbohydrate? I'm going to talk about Krebs cycle, gluconeogenesis, and I will review some point again regarding to the glycolysis regulation and gluconeogenesis. At first, I'm going to talk about Krebs cycle. I think you are familiar in your high school about the Krebs cycle, yes?
Yes.
In a Krebs cycle, actually, the purpose, very in a simple word, produce the energy, okay? So you have some intermediates. Do you know what's the meaning of intermediate? Actually, intermediate, that they are full of energy. At first, acetyl-CoA, for example, most of them result from the actually carbohydrate. You should know that other molecule can produce them. Acetyl-CoA, as a result of the pyruvate dehydrogenase reaction, combine to the oxaloacetate and produces several compounds. And I will review one by one. Many years ago, Krebs was a scientist that introduced this pathway, and we have several names. Krebs cycle, because the first product is the citric or citrate, the name of the cycle, now as a citric acid cycle, or because you have some compound with a 3 carboxyl group. Okay, so other name is a tricarboxylic acid cycle. But you should know that during the Krebs cycle, we cannot say this is an anabolic or catabolic, okay, purely. It is a amphibolic. Amphibolic, it means that both of the oxidation as a catabolism and photosynthesis as an anabolism occur inside the Krebs cycle. It has a very important role, especially central role, in the metabolism of the biomolecule. Like, for example, because of the oxaloacetate, it is important in the gluconeogenesis. And also because of the acetyl-CoA. It is important in the metabolism of the lipids. And so this is very, very important cycle. About the localization, where is the site of Krebs cycle? Mitochondria. Okay, so you should know this is not any site there, cytosol. About the reaction, this is an overview of the Krebs cycle. At first, oxaloacetate combined to acetyl-CoA by the name of the enzyme is citrate syntase. Convert to the citrate as the first product. After aconitase, isocitrate produced, and after that, by the action of isocitrate dehydrogenase, the first NADH is produced. Convert to the alpha-ketoglutarate. Please tell me about the number of the carbon. How many carbon present in oxaloacetate? What about acetyl-CoA? Two carbon. Pyruvate convert to acetyl-CoA after the carboxylation. Pyruvate has a 3 carbon, acetyl-CoA 2 carbon. What about the isocitrate? 6. What about alpha ketoglutarate? 5. Because the first carbon is absent. What about alpha ketoglutarate? 5. What about succinyl-CoA? 4. And in continuous succinate, fumarate, malate, all of them are 4 carbon. As you can see here, there is no release of CO2, clear? So whenever you have a decarboxylation, it means that the number of carbon change, okay? If it is 5 carbon, it's converted to the 4 carbon. If it is 6 carbon, it's converted to the 5 carbon. So, I want to ask a question about this chart. How many decarboxylation reaction is present? Okay. How many NADH is produced? How many FADH is produced? I do apologize. How many GTP is produced? 1. So for the calculation of energy, you know 1 GTP in some references consider as a GTP because it's a high energy. Exactly like it, ATP. Okay? So 1 ATP. 3. Number 1, number 2, and number 3. 1 NADH2 is present and 2 CO2 is released from the reaction. In the next slide, I will review one by one. As I mentioned, the link reaction between glycolysis and Krebs cycle was pyruvate dehydrogenase or pyruvate decarboxylase. You are right. Because both of them are correct. Some part of the enzyme has a dehydrogenation function, some part has a decarboxylation. Both of them are correct. But pyruvate carboxylase is different. Okay? Please do not make a mistake. Pyruvate decarboxylase, pyruvate carboxylase. When you add a 1 carbon by the help of pyruvate carboxylase, it's converted to oxaloacetate. After the carboxylation, pyruvate converted to decarboxylation. Acetyl-CoA. Okay, I want to repeat it a lot to memorize in your mind. As I mentioned, pyruvate has a specific novel here. Okay, sent to mitochondria. In mitochondria, convert to acetyl-CoA, and acetyl-CoA in turn to the Krebs or citric acid cycle. As I mentioned, because citrate is the first product, the name of the cycle now as a citric acid. Citric acid cycle, or because of the discoverer, Krebs cycle. And as I mentioned about the localization, you should know that it is not not inside the cytosol. It is in the mitochondria. This is a review of the pyruvate dehydrogenase because I want to start with the acetyl way. As a result of conversion of pyruvate while the dehydrogenase convert to the acetyl way, and as I mentioned, this is a large complex. The detail is not important, okay? I never ask you about the detailed part of the pyruvate dehydrogenase. I just put this slide because I want to show this is not simple, very large and complex enzyme. Okay, very and highly regulated. I want to review one by one the reaction of the Krebs cycle. After condensation, condensation means ترکیب in Farsi. Do you know what's about condensation? ترکیب in Farsi, again, alhamdulillah, we have very common word. Condensation of the acetyl-CoA and oxaloacetate citrate produced. At first, I want to direct your attention to this arrow. It is reversible or irreversible? Irreversible. So it is regulatory. So if I ask you, what is the first regulatory enzyme in Krebs cycle? It is citrate synthase. And it can inhibit by its products, exactly like a feedback inhibition. So this is the product, so it inhibits it. Sorry. This is inhibit citrate synthase. As you can see here, when we say tricarboxylic acid cycle, this is the other name of Krebs cycle. Tri means 1, 2, 3, COO minus. It means that carboxyl group, okay? When we say this is a tricarboxylic acid cycle, it is because of during this cycle we have it 1, 2, 3 for this cycle. About another step, after production of the citrate, by the help of aconitase, it converts to the isocitrate. This is a reversible reaction, so it is not a regulatory enzyme. And after that, isocitrate convert to the alpha ketoglutarate. How many carbon present in isocitrate? 6. What about alpha ketoglutarate? 5 carbon. As you can see here, this complex is similar to the pyruvate dehydrogenase. In a pyruvate dehydrogenase, also we have a decarboxylation. Can you see here? In pyruvate dehydrogenase, NADH produced, in isocitrate, NADH produced. In pyruvate dehydrogenase, CO2 release. In isocitrate dehydrogenase, CO2 release. About the regulation is very important. About the ATP, it is clear. If you are in a good mood, high energy, I mean high ATP, it can as an activator or inhibitor? Inhibitor. What about ADH? ADP? Activate, because it's ADP means that the level of energy is low. What about NADH? NADH considered as a 3 ATP, so it means a high energy. Okay, so it suppresses, inhibits the Krebs cycle. What about the calcium? You should know that for the enlabazat azole, okay, muscle contraction will require when you are in a vigorous exercise, you need energy. The calcium increase, okay? When we need energy, calcium increase. So when you have the calcium, it acts as an activator. So, for any process or pathway that produce energy, calcium is activator. Okay? Why? Because calcium show that you have an active muscle. You are in a very intense exercise. So it show that we require energy. So it's considered as an activator. After that, conversion of alpha-ketoglutarate, after that, isocitrate convert to the alpha-ketoglutarate. Alpha-ketoglutarate convert to the succinylate. How many carbon present in succinylate? 4. So, from this point up to the remaining cycle, we have a 4 carbon compound. Again, alpha-ketoglutarate, again, it's example like a previous enzyme. This is a large complex. As you can see here, CO2 release, NADH release, and about the regulation, calcium activates, NADH inhibits, and what about the product? It is a feedback inhibition. Okay? Please do not actually try to memorize, just understand. When you have a high substrate فیلم نگاه میکنه ها خدا شاهده. I want to change my face. Watching a movie? What was that sound? Okay. بزدنش بیرون؟ به من نمیاد. I cannot be angry. It's not in my nature, unfortunately. Unfortunately. As you can see here, okay, I will try to speak word by word. Okay, I do apologize for the student. They can't understand English. Okay, this is for a student. Maybe they have some problem with writing. Alpha-ketoglutarate convert. Is it good way? No. You understand? You really? Okay. Alpha-ketoglutarate convert to the okay, I will try to control my as I mentioned, whenever we have a large complex of the dehydrogenase, it means that it requires more than one coenzyme, for example. But in this slide, I showed just 2 of them. One of them is CoA, because alpha-ketoglutarate, is there any bonds in A? No. What about this one? Succinyl-CoA. So if I ask you, what are the coenzyme for alpha-ketoglutarate dehydrogenase enzyme? These are CoA and also NAD+. Okay? This is the site of the production of NADH. So, during this reaction, NADH produced after decarboxylation. Coenzyme, NAD plus. Okay, this is not NADH. NAD plus and coenzyme. Okay, for this reaction. Exosuccinyl-CoA produce. If you remember, I mentioned this point. Whenever you have a CoA, it means that this is a high energy molecule, like acetyl-CoA. چی شد؟ اشکالی نداره. Acetyl-CoA. Acetyl-CoA contain high energy. Succinyl-CoA. As I mentioned, CoA means high energy. Okay? So, succinyl-CoA, because contain a high energy, it can release its energy in a form of GTP. Clear? So, this is a very high energy molecule. It can release its energy in a form of GTP. You should know that we have one expression. This is a substrate, the yellow box. Substrate level phosphorylation. What does it mean? It means that whenever the source of the energy is a substrate that is not ATP, this is a substrate. This is not ATP. Sometimes ATP donates its energy to convert to ADP. Okay, this is not this reaction. Then you have a substrate that is full of energy to produce one GTP or ATP. It's known as a substrate level phosphorylation. Another reaction is a conversion of succinate to the fumarate by the succinate dehydrogenase. Are you familiar with the respiratory chain? I mean electron transport chain. Are you familiar?
Yes.
We have actually inside the mitochondria, inside the disease, MIT is abbreviation of the mitochondria. Okay? Inside the mitochondria, in a very, very simple word, we have some complex. Complex number 1, 2, 3, 4, and finally 5. In a 5 complex, ATP produced. Okay? From complex number 1,
NADH.
NADH. NADH enter from complex 1. From complex 2, FADH enter. Okay? The name of the complex 2, I'm talking about this complex, is succinate dehydrogenase. This is exactly this enzyme. Okay? Succinate dehydrogenase is the name of the complex number 2 of the ETC, electron transport chain. Okay? And it contains a very, very important point, and inshallah, Dr. Panahi studied it. After that, you have a hydration of the fumarate, like the fumarase modulate is formed, and dehydrogenation converts to the oxaloacetate. At the end of the cycle, as you can see here, the third, number 3, okay? The number 3 of NADH is produced. As you can see here, the third, and in other words, the last NADH produced. So, if I ask you the name of the enzyme that for the production of last NADH, it is a malate dehydrogenase. This is an overview of the citric acid cycle, about the site of the production of the coenzyme. These are in biochemistry, actually they have expression for NADH and FADH. These are known as a reduced coenzyme. Okay? We say reduced. Reduced means that they are in a reduced form, or they have a hydrogen atom, or in other words, they have a high energy. As you know, they insert to the ETC, electron transfer chain, and they release their ATP. For each NADH, how many ATP produce? 3. For each FADH, how many ATP produce? 2. So these are a high energy compound. So if I ask you about the energy calculation of the TCA, overall 3, 1, 2, 3. Consider as a actually 9. Do you know why?
Yes.
Because 3 times 3 is 9. 1 FADH2 produce, consider as a 2 ATP. And 1 GTP consider as a 1 ATP. So the total energy production is 12. If I ask you how many ATP produced by the entering of 2-acetyl-CoA nucleoside? 24. How many ATP produced from entrance of 1-acetyl-CoA? As I mentioned, 2 pyruvate is complicated. I didn't want to confuse you. Okay? As I mentioned, any products containing CoA has a high energy. As you can see here, 1-acetyl-CoA produces 12 ATP. So it means that acetyl-CoA has a very contain high energy, and step by step release its energy. I want to review the energy molecule. As you can see here, if I consume, this slide is very, very important about the calculation of the total. I mean that because I show the glycolysis link reaction. What was the link reaction? Pyruvate dehydrogenase or pyruvate decarbonate. It is most common to say that pyruvate dehydrogenase, okay? Because in abbreviation it is PDH, pyruvate dehydrogenase, okay? This is the overview of glycolysis. I'm talking about aerobic. Please pay attention. If you have anaerobic, the calculation changes. Okay? You should consider the conversion of pyruvate to lactate. But now I'm talking about aerobic. From glucose to pyruvate, as you can see here, 6 ATP by the help of 2 NADH. Do you remember? ATP product. About the link reaction, this is a linker between glycolysis and Krebs cycle. 2 pyruvate, 2 NADH. So 2 NADH, 6 ATP considered. What about Krebs cycle? As you can see here, after 2, okay, 2 acetyl-CoA, 18 ATP here, 4 ATP, 2 ATP. If you actually consider all of these ATP, already about 38 ATP is produced in this reaction. This is a very high energy.
So, if this is that, what is the
Anaerobic, what happened? Pyruvate, I wrote the abbreviation, convert to lactate. For one pyruvate, one NADH is required. One NADH is required. If you have two pyruvates, two NADH is required. You should pay. Two NADH means that how many? Six. So 38 minus 6 is 32 in an anaerobic situation. You understand?
An aerobic situation will include the carbon Krebs cycle.
Anaerobic can insert. How they can produce, for example, RBC, it should stop at the glycolysis? No. After production of the lactate, actually, is a reversible reaction. Please pay attention, The reaction of the lactate and pyruvate is reversible.
Yes, but there is no mitochondria, and this Krebs cycle happen in the mitochondria.
Oh, yes, you are right. I make a mistake. What are you talking about? Krebs cycle is not present in RBC. Yes, you are right. I'm really confused. I'm talking about this, for example, some, vigorously exercising muscle. Okay? This reaction, again, here, there actually we have it anaerobic glycolysis. But muscle has a mitochondria. So you have a reverse reaction in the muscles, pyruvate convert to the acetyl-CoA, and acetyl-CoA enter to the Krebs cycle. But please pay attention, refer to the good point many times. Please pay attention whenever I wrote the name of the cell, I mean the RBC or muscle, you should know that RBC don't have any mitochondria. But if I ask you anaerobic, maybe it means that you have an anaerobic muscle. Okay? So you should consider the point many times for mentioning good point. Whenever you actually want to calculate the ATP in a RBC, there is no Krebs cycle. So up to here is block. Okay? So please pay attention. In RBC, pyruvate dehydrogenase present or not? Not. Okay? So in RBC, because mitochondria is not present, so pyruvate cannot convert to the acetyl-CoA. So it converts to the, it catalyzes a reverse reaction. It catalyzes a reverse reaction of the pyruvate to the lactate, and from that way it can produce more energy. Repeated glycolysis inside the RBC. But in an anaerobic, like a muscle, pyruvate converts to acetyl-CoA, and in anaerobic muscle, you have a Krebs cycle. Okay?
Absent in the RBC.
Exactly. About the enzyme, in this slide, I give you the name of the enzyme, aconitase, isocitrate dehydrogenase, alpha-ketoglutarate. These three boxes is important. You should, if I ask you about the name, you should memorize the name of the regulator enzyme. Number one is citrate synthase. Number two, isocitrate dehydrogenase. And the last one is alpha-ketoglutarate dehydrogenase. About the regulation, as you can see here, citrate as a product feedback inhibition. Clear? It can inhibit this enzyme because citrate synthase combine oxaloacetate to acetyl-CoA, produces citrate. As I mentioned, when citrate is high, Krebs cycle is suppressed or inhibited. What about other enzyme? One by one, I want to review. Krebs cycle produce energy. So, NADH inhibit or activate? Inhibit. Because NADH, it means that I'm in a good mood. What about ATP? Inhibit. What about ADP? Oh, I'm hungry. Actually activate the cycle. What about calcium? Activate. And about this complex alpha ketoglutarate, exactly like NADH, and calcium is activate. And as you can see here, this is a product feedback inhibition. Product inhibit enzyme. Okay? All the time. Substrate. Please pay attention. This is enzymatic reaction. Am I right? Substrate. Convert to product. This is the enzyme. Do you remember from enzyme session? Substrate, convert product. So, all the time, substrate activates enzyme, product inhibits enzyme. Okay? This is a general rule. This is a rule in biochemistry. Enzyme activated by substrate inhibits by product. Like here, succinate-CoA is product, so it inhibits enzyme. Clear? I'm good at this. About the localization, I want to compare it. As I mentioned, in RBC, we don't have this reaction, pyruvate to acetyl-CoA, Krebs cycle, and respiratory electron transfer chain. These pathways are just found in cells that they have a mitochondria. Okay? So whenever you don't have any mitochondria, you stop here glycolysis and actually send into the other pathway. But citric acid and respiratory electron transfer chain is just for the cells, for the mitochondria. Now the gluconeogenesis, I will review the principle, and after that, after 2 slides, because at first I want to show what happens to the body if we are lack of glucose. I mean that you should know that some tissues, like brain, like RBCs, kidney, lens, some parts of the eye, they require, or vigorous muscle, they require continuous supply of glucose. Who provide the glucose in a normal situation? What is the storage form of glucose? Glycogen. But if you have a prolonged fasting, very actually long fasting, what happens? You should know that. Where is this site of storage of glycogen? Liver and glycogen.
Liver and muscle.
You are talking about gluconeogenesis. Okay? If I ask you what is the site of a storage of glycogen, it is inside the liver and muscle. But you should know that liver glycogen can provide the energy less than 1 day. Okay? In the absence of the dietary carbohydrates, especially, for example, you are in a fast more than 1 day, It's abnormal, but sometimes happen, okay? More than 1 day, 20 hour, more than 1 day fasting, actually the hepatic glycogen store completely used, okay? So, but we require glucose. How? By the gluconeogenesis. As I mentioned, from the non-carbohydrate precursors like a pyruvate, like a lactate, like a glycerol, and alpha-keto acids. You should know that gluconeogenesis is not a, in a simple way we can say that glycolysis, gluconeogenesis, okay? But it is not very simple. We have some enzymes that are unique to the gluconeogenesis. Do you remember the 3 enzymes
Yes.
that irreversible in the glycolysis? Please review the name. Hexokinase, fructokinase, and pyruvate kinase. So this 3 reaction is different in a gluconeogenesis. And you should know that there is the site of the localization of gluconeogenesis in a cytosol or mitochondria. No. Gluconeogenesis happen
In both.
both of them. But you should know that one exception. If substrate is glycerol, please pay attention, maybe I ask you this point. If substrate is glycerol, the location is just cytosol, okay? But about the other compound, both of them are involved. Deficiency of this process causes a very clear hypoglycemia. What does it mean, hypo?
Low
Low
concentration.
concentration. You're near to faint, okay? Because our brain, and during the normally overnight fast, 90% occur in the liver and 10% kidney. What if you continue fasting, continue, the percent of the kidney increase? This is the overview of gluconeogenesis. As you can see here, glucose when converted to the pyruvate, the pathway is glycolysis. But the reverse pathway is gluconeogenesis. We have a 1, 2, 3, 4 unique reaction. If I ask you how many reactions is common between, completely common between glycolysis and gluconeogenesis? 7 reactions.
Reversible.
Because these are reversible. So 7 reactions are actually reversible. Irreversible actually, so they are unique. As you know, hexokinase, phosphofructokinase, and pyruvate kinase are actually irreversible. But 7 is reversible. 7 reaction reversible, but 3 reaction in a glycolysis is irreversible. So you should bypass this pathway. Bypass means that turn, make him leave, okay? Bypass. By the help of some unique enzyme. It's better to have a rest, but I will start.